I'm Erez Shor. I'm the CEO and co-founder of Exero Medical. I'm an engineer and scientist by training with some startup experience, but I wanted to talk to you about my life. Do I have a clicker? All right. You see? I think it was a prime example. This is my life. I don't know anything. I wake up in the morning. I don't know if the shipping will come in time, if we are able to enroll those patients, what's going on with the study. This is my life. But I think that I'm not alone. Any surgeons in the audience? I thought so. I'm complaining about my uncertainty, but I think you are in a completely different situation. So here is the father of our CFO, undergoing surgery to remove a tumor, rectal resection, circular anastomosis, and the operation goes uneventfully. And then after 2 days, you don't know. It's this uncertainty. So the surgeon sits tight. After 4 days, the contrast CT comes back negative, and he still suspects, the surgeon, that something is wrong there. So the patient is diagnosed with an anastomotic leak and undergoes the catastrophic sequela of revision surgery, a couple days at the ICU, longer hospitalization, the story, I guess, of your life. Now, of course, this patient is not alone, and this impacts millions and millions of patients worldwide. The complication rate is high, the burden to the healthcare system is high, and the mortality is staggering. And throughout the past few decades, many attempts were made to prevent those complications by better staplers, by buttresses, by glues, by all sorts of devices, but nothing proved effective enough to prevent those complications. And so we thought maybe the complications cannot be avoided, but maybe the catastrophic sequela can. And so we designed a sensor that actually interfaces to the surgical site and is able to monitor some crucial physiological processes underlying the complication, underlying the leak. And so the clinician can have this window of opportunity to intervene before the catastrophe has manifested. The sensor is embedded in a standard surgical drain, so we don't change the surgical procedure at all. We don't change the patient's workflow at all. The data collected from the sensor is relayed using a small wearable to a secured cloud, and from there, indications can be given to the clinical team. I'm saying physiological processes. We're interested in continuous monitoring of inflammation. We expect inflammation after surgery, but we also expect this inflammation to subside. In other cases, if it doesn't, then there is a problem. We monitor for ischemia or ischemic hotspots during or around the anastomosis, and we are monitoring for overall motility. And the combination of those three parameters is indicative of a problem. Ever since we started, we went through the translational studies, starting with small animals, large animals, ex vivo study with functional colonic human tissue. We did a first-in-human just to show that our working hypothesis is working. And then we continued to a pilot study in which we enrolled 50 patients in 6 medical centers in Israel. We enrolled 50 patients. The patients must have read the literature because 6% of them had a catastrophic leak that required a revision surgery. And the device was able to detect those patients on postoperative day 3 with 100% sensitivity and 100% specificity. As a matter of fact, if we're looking at how this data is handled, each patient is represented in this chart by a single point, and the 3 parameters I referred to are converted to this 2-dimensional space. And as you can see, the patients with a leak feature low motility, high inflammation, and those hypoxemic hotspots, unlike the patients who did not develop the leak. And if I compare it to the standard of care, I think the real problem is you will have some false negatives with your contrast CT because there is a problem, but you don't have an actual leak yet. But those processes are there before. And so if you compare the sensitivity and specificity of XBAR versus CT, you see that on postoperative day 3, this small cohort of 50 patients yielded 100% sensitivity and 100% specificity. On postoperative day 2, It was at 100% sensitivity and 92% specificity. So real actionable data in a clinically viable time. Of course, the data we collect is quite rich and can be developed into additional applications other than colorectal surgery, of course, and also for other parameters of colorectal patients. But I think the picture of what's in it for the patients, for the surgeons, for the providers is wider. So for the patient, it's straightforward. Better outcome, shorter hospitalization. I think surgeons can also benefit from better sleep at night. And if I look at providers, then of course we have reduced cost and better outcomes overall. Also, this is already covered in the existing DRG reimbursement, so the hospitals can exploit this right away. And it's applicable for NTAP. So again, the hospital may be completely agnostic to the price of the device. But even if the hospital is not, if we look at the price point of today versus better post-op monitoring, today typical patient costs more than $16,000 for the hospital. And that's a result of the long hospitalization and of course the tremendous cost of a leak. If we were able to shorten hospitalization for both populations, if a patient is doing well, you can do way more aggressive enhanced recovery and send the patient back home earlier. If the patient, however, is likely to develop a complication, you can intervene and prevent the catastrophic sequela, save time, money, and lives. $4,000 per patient, and if we're looking at a large center doing 1,000 cases a year, we see a very considerable savings in lives and also in money. We are actually moving ahead very quickly. We have a breakthrough designation, and we have the pivotal trial that we commenced a couple weeks ago, approved by the FDA. Our regulatory pathway is a de novo clearance, so we can estimate the time between the end of the pivotal study and the actual clearance. And I'm also happy to say that 2 months ago at NBT, I said we plan to commence our pivotal study. Now we commence our pivotal study with 5 sites already activated, 3 in Israel, 2 in the US. We have 12 patients already in, and it's moving along quite nicely. And if I'm looking at the overall market, so the market for drain-based applications is around $3 billion per year, looking at the volume of surgeries. We know that you don't use drains in all indications and in all cases, and for that, we have Gen 2, which features the same sensing device, but embedded on a resorbable raft. So we can place it during surgery and have the same monitoring and use it on patients where drains are not commonly used. We are Exero Medical. We are very lean. I think also we're quite mean. We're doing prevention and monitoring of anastomotic leaks. That's what we do. That's what we think about. That's our profession. A small team of doctors, of scientists, of engineers, of clinicians, of business persons, and we are here to give certainty. This is about giving certainty. So now it's your opportunity to join our journey. The easiest thing you can do, of course, is to vote for us. But being serious for a moment, as I said, we started our pivotal study. We would love you to reach out and see if you can be a part of this study, if you can be a part of how this can impact your health system. I'm also happy to report that we've been working on our fundraising, and we're very close to closing our seed round of $8 million overall. I just was working on the definitive agreement this morning before I came over. And with that, I would like to thank you for your attention, invite you to really reach out and see what's in it for you. And I'm happy to take questions, Sharks.
So what are you actually measuring? I mean, you talked about motility and this and that, and it seems like there was some smoke and mirrors to get to 100% sensitivity and specificity in 3 days. I appreciate the fact that there may be stuff happening before the most leaks appear between day, 7 and 10, depending on, but what are you measuring?
Can you hear me?
Yeah.
Okay, so the sensor is based on electrophysiology. We have an electrode array that is embedded throughout the drain, and this electrode array serves for three types of parameters we're measuring. The first one is inflammation leads to secretion of factors that in turn change the electrical conductivity of the tissue. So we measure the electrical conductivity of the tissue, and that allows us to see how the inflammation of the tissue develops. Another parameter we're measuring is the slow wave frequency of the digestive tract in cycles per minute. So this is the most straightforward parameter to measure motility. And the third parameter we were measuring, you can consider this electrode array as something very similar to a distributed EKG system. So we have a couple electrodes lined along the digestive tract, and we actually measure the spiking activity driving peristalsis. The spiking activity driving peristalsis is indicative of ischemic hotspots, because once you're able to compare what's happening on an intact tissue versus what's happening near the anastomosis, and they're different, you very easily can identify ischemic problems. So those three parameters, impedance for inflammation, frequency for motility, and then spiking activity for ischemic hotspots are the parameters we're measuring.
So you talked about false negatives with the CT scan. I guess I'm concerned about false positives. So, for example, an appendectomy, you almost never have an anastomotic leak. You have a pretty high incidence of abscesses from contamination. Is this really to identify infection, or is it to identify a leak? What is your target here?
Our target is actually to identify those patients that are likely to require an invasive procedure to solve a leak. And I'm not saying to identify a leak, because I think a leak is when it's already too late. So we're looking for those patients. Now, if the patient only shows very high rates of inflammation, but his motility and overall spiking activity are okay, we would not classify this patient as a patient that is likely to develop this complication.
Yeah, but it's not that clean, right? So if you get an abscess and you don't have a leak, but you have an infection, many of those patients get an ileus and have poor function as well.
So going back to how we calibrate how we calibrated the algorithm, there is some debate about the definition of how to classify leaks. And if you go to the original classification that says that a severe leak is only major surgery, but then if you need to have your interventional radiologist drain an abscess, it's classified as leak grade B. We, in our endpoint, are looking for classifying those leaks that would either lead to a revision surgery or lead to some invasive interventions, such as this radiological draining of an abscess. And this is the endpoint of our pivotal study to the FDA. We need to show our performance in detecting leaks that we defined as severe using this definition.
So I'll pick up from there at a different level. And I'm going to bite. I really am. Before I do it, I want to congratulate you on your work. I mean, I think I'm a sensors person. Anytime you put a sensor anywhere, I think it's really cool. It doesn't even matter what kind of sensor it is. I just love it. And congratulations on your money and your raise. Here's your problem. Surgeons don't believe anything you say. I mean, in order to get us to change our workflow that we worked really hard on to perfect, you have to show us the data. So she gave you a data bite. She bit your leg, and it was because she wants to see the proof, the data, something that's gonna make us change our workflow. And so that's the pie in the sky. So I have a million questions I'm not going to ask you, but in what we don't understand is how does this truly fit in the clinical workflow? And we're visual, so you have to give us a presentation that says, here's our thing in your drains that you already put in. What drain company? I'm just going to give you the questions. I don't want you to answer them right away. We want to know what drain company you're working, because we're not going to change our drains. Some of us love our Blake drains or whatever kind of drains. I mean, those companies are going to have to integrate it. Maybe you have a completely new drain that has your sensor in it already. That's problem number one. We already love what we have in terms of just regular old-fashioned drains. That's problem number one. Problem number two, I want to know from your perspective, the studies that you did that gave this kind of indicator, whether it was inflammation, whether it was ischemia, motility, like to what level, show us the data. We're scientists. In God we trust, the rest of us must have data. And we're wanting the data. We're asking data questions. So we don't understand, what do we see that then makes us act and then takes us to the abscess pathway or back to the operating room? I'm going to go back to the operating room with the data that comes out of your sensor. Those are the...
Good questions. Actually, you mentioned I have two problems, so I will try to address both. The first one was how the device looks like and get you some tangible feeling of how it looks. It looks like a standard Jackson Pratt drain. So I think it's featured in one of the images, but I'm happy to follow up on it. The electrodes we embed in the...
Flat, round, French size. Can I cut it?
You can cut it. It can be easily adapted to other types of drains, but it was designed to show that this thing is viable. The translation to Blake drains, the translation to other size drains is relatively straightforward. Each electrode is 1 millimeter by 300 micron diameter, so the electrodes are small and can easily integrate it into different types of drain. We started using flat JP drains because these were common with the surgeons we worked with. Now, for the data, I actually not only agree, but I'd be more than happy to dive into the entire translational process of how the raw data looks like and how can we actually infer physiology from that data. Now, when you talk to surgeons, by the way, and we did a surgeon survey about the usability of the system, and this is a process that is in progress, it's interesting because some surgeons say, don't bug me with data, I just need a traffic light. I want to have a red, amber, green, that's good for me. Don't annoy me with trends of different parameters you're measuring. And others are saying, we would like to see the data. We would like to see if we give prophylactics, whether this inflammation subsides or is worsening. So it's
But
super...
you're confusing, operability with acceptance that the red means something bad and the green means something good. I agree that red, yellow, green is what they want to see once they believe that it's valid. And so I think that's part of the problem. If you don't have ground truth about the leak, which I think a little bit is what Danielle was talking about, you can't just use your own data as we said you had a leak, and that happens 6% of the time.
So let's talk about ground truth for a brief second. The study is designed to support this ground truth. So in the study, the clinical team is completely blinded to the results of the device. We collect the data. The data is processed independently, and retrospectively, the outcome that was actually defined by the clinical team is compared to what the device did. So we don't have any magics about manipulating the data. And I'm happy to follow up and actually dwell into how the raw data looks like and why this is representative of the physiology. Some proceedings were already presented in journals and in conferences and are available to review.
But we'll all look forward to that data, Dan.
We often hear in our morbidity and mortality conferences about surgeons who put Jackson Pratt drains right next to their anastomoses and fail to detect a leak, either because the drain became encapsulated. We know that silicone drains very quickly become encapsulated in a dense fibrous container, if you will. They also slip out of position. What is to prevent the same from happening to your sensor?
So the sensor is capable of being there and monitoring for a relatively short amount of time, and we didn't want to change the workflow in any way. So the sensor is placed and then sutured to the skin. Again, no change to the standard procedure. In our study, we verified that the drain doesn't move using abdominal X-ray before the patients were discharged, and we verified that the drain hadn't moved. We didn't have any encapsulation problem in 2-3 days following surgery.